Hey everybody, we are going to continue on with this unit and we're going to start today talking about meiosis and the phases of meiosis. We're also going to look at the differences between meiosis and mitosis. So mitosis was one cell division, whereas meiosis is going to have two cell divisions. And because you're having twice as many cell divisions, you're also going to have twice as many cells produced. So at the end of meiosis, we're going to have four cells that are produced. We'll also see something called a synapsis and chiasmata that will be observed in meiosis. So with this first division, you're separating pairs of chromosomes, not duplicate chromosomes. And you have a phase called interkinesis that is also present. So like I said before, meiosis has two cell divisions. So the steps that you follow have the same names as for mitosis, but you add a 1 or a 2 to the end of the name, and that's how you kind of figure out the phases. So you had prophase, metaphase, anaphase, telophase for mitosis. With meiosis, you'll have prophase 1, metaphase 1, anaphase 1, telophase 1. Then prophase 2, metaphase 2, anaphase 2, telophase 2. So here we're looking at interphase 1. You would have sort of like the time before the division takes place. You have a homologous pair of chromosomes in that parent cell. So, during interphase, you'll have the DNA replicate, and so now you have your sister chromatids. And so now you're looking at a homologous pair of replicated chromosomes. So the one in blue could represent the chromosome that you inherited from dad, and then the one in red could represent the chromosome you inherited from mom. And so the first thing we need to do is separate those homologous chromosomes. Once they are separated in meiosis 1, then it'll be meiosis 2 where you separate the sister chromatids. Okay, so we have to kind of add that extra round of division because we want to have half as many chromosomes at the end as opposed to having a complete set of chromosomes like you want with mitosis. So with prophase 1, the basic step is the same as that of prophase in mitosis. But you also have synapsis occur as the chromosomes condense, and this is when the homologous chromosomes form what we call tetrads. And so that is kind of what we're looking at here. And so this would be a tetrad. These are homologous chromosomes again, so this would be like chromosome 1 from dad and chromosome 1 from mom. Because they're both chromosome 1, they are going to line up together and form that tetrad. Also during prophase 1, you have the chiasmata observed. This is also going to be the longest phase of division. With metaphase 1, the tetrads are going to align on the metaphase plate, and then the centromeres of the homologous pairs are going to point toward opposite poles. And then with anaphase 1, the homologous pairs are going to separate, and now the duplicate chromosomes are still attached at the centromeres. So the sister chromatids are still attached, however the homologous pairs are separate. This is just showing you some of the possibilities for anaphase 1. And what I mean by that is if we're looking at two sets of homologous pairs, there's always the possibility that both blues could get pulled to one side and then both reds could get pulled to one side, or there's a possibility that a blue and red would get pulled to one side and then a red and a blue would get pulled to the other. So there's different possibilities for anaphase. So what happens during anaphase 1 is that the maternal and paternal chromosomes are now separated randomly. Okay, so that's why we're showing two different colors. So the blue would be chromosomes inherited from dad, the red would be chromosomes inherited from mom. They don't have to stay together in terms of like all the blue together and all the red together. They're going to mix up a little bit, and that's actually really important for genetic variation. Now in telophase 1, again, this is very similar to mitosis. And so the chromosomes may or may not unwind to chromatin. Cytokinesis is going to separate the cytoplasm, and you'll now have two cells that are formed. So here we're going to kind of look at all of this together. And so we have prophase 1. So the same kind of things that were happening in mitosis are happening now. But you have the homologous chromosomes forming the tetrad. So here's a homologous set, here's a homologous set. And so you can see that they're kind of pairing up together. You have the spindle forming, and you have the nuclear envelope breaking down. Then during metaphase 1, the homologous pairs are lining up along the metaphase plate. During anaphase 1, you have the homologous chromosomes being separated. And now in telophase 1 in cytokinesis, the cell is starting to split in half. You have your nuclear envelope starting to reform, and you have your homologous pairs that are completely separate and a nucleus starting to form around them. This is just kind of zooming in on all of that. Then we have interkinesis. Interkinesis is sort of the phase in between meiosis 1 and meiosis 2. This is not going to be a time for DNA synthesis. So when we talk about interphase happening in between each round of mitosis, DNA is always synthesizing in that time. This is not happening here because you want to reduce the number of chromosomes by half into each of the new daughter cells, so you don't want to replicate your DNA. Interkinesis could last for a long time, or a cell could go immediately into meiosis 2. It kind of looks similar to interphase of mitosis. And then after interkinesis, you have the steps of meiosis 2, which are the same as in mitosis. So again, we have prophase 2, so the nuclear envelope is breaking down, the spindle is forming. In metaphase 2, the chromosomes are lining up along the metaphase plate. In anaphase 2, you now have the sister chromatids being separated, and you can see that this is all happening in two different cells at the same time. And then you have telophase 2 in cytokinesis. So again, telophase 2, you have the chromosomes unwinding into chromatid. You have the nuclear envelope reforming, and during cytokinesis, the cells are starting to split in half. So you can see that you are going to now have 4 daughter cells as opposed to just with the single division of mitosis where you only have the 2 daughter cells. So the results of meiosis, again, you have 4 cells produced. The chromosome number is halved, and this is how you're making your gametes or your sex cells, so eggs or sperm. And genetic variation is increased because you are separating the homologous chromosomes randomly, so you could have some from mom and some from dad. You also have crossing over occur, so you're definitely increasing genetic variation. So your lecture question for today is, in your own words, can you explain why meiosis needs 2 rounds of division as compared to meiosis, which only requires 1 round? All right, guys, don't forget to send me a message via the class website with your answer to the lecture question. Thank you.